Welcome back to Antibody Basics by Biointron Part 5. In this episode, we'll continue to cover antibody formats, specifically Fab, Fab2, and Fc fragments. Antibody fragmentation is the biochemical or genetic cleavage of antibodies into smaller fragments. This process is used in various laboratory and clinical applications, as fragments retain the specific targeting capabilities of full monoclonal antibodies while altering physiochemical features. There are several different types of fragments available, such as small monovalent antibody fragments, Fab or scFVs, and engineered variants, diabodies, triabodies, minibodies, and single-domain antibodies. The fragment antigen binding region is typically formed by enzymatic cleavage of the antibody molecule with papain, resulting in two identical Fab fragments. It contains the variable regions of both the heavy and light chains. Each Fab fragment contains one antigen binding site and is responsible for binding two antigens. Fab2 results from enzymatic cleavage of the antibody molecule, but with pepsin instead of papain. Fab2 fragments consist of two Fab portions connected by disulfide bonds in the hinge region of the antibody. This fragment lacks the Fc region and is bivalent, meaning it can bind to two antigen molecules simultaneously. The Fc region is the crystallizable fragment of an antibody. It contains the constant regions of the heavy chains and is involved in various effector functions, such as antibody-dependent cellular cytotoxicity, ADCC, complement activation, and binding to Fc receptors on immune cells. The Fc region plays a crucial role in the immune response and antibody-mediated clearance of pathogens or target cells. The emergence of antibody fragments began in 1949, when Rodney Porter introduced the concept of enzymatic digestion with papain to cleave antibodies into Fab fragments and an Fc region. In the similar times, Alfred Nisonoff used pepsin on rapid IgG and observed the generation of a single bivalent fragment Fab2. In 1972, Rodney Porter and Gerald Edelman won a Nobel Prize for independently determining the basic structure of immunoglobulins, revealing the presence of two identical heavy chains and two identical light chains. By the 20th century, advances in genetic engineering and recombinant DNA technology enabled the production of engineered antibodies with modified Fab, Fab2, and Fc regions, leading to the development of therapeutics. And from the 2000s to the present time, there are continued R&D efforts focusing on optimizing stability, specificity, and pharmacokinetics, leading to the emergence of novel formats such as bispecific antibodies and antibody-drug conjugates. The most common method of producing Fab, Fab2, and Fc fragments are via enzymatic digestion, with proteases such as papain, pepsin, and physin. First, we can start with papain digestion. Papain is a cysteine protease which specifically cleaves peptide bonds adjacent to amino acids with bulky hydrophobic residues such as phenylalanine, tyrosine, and tryptophan. In IgG, papain cleaves the hinge region between the Fab and Fc fragments, resulting in the formation of two identical Fab fragments and an Fc fragment. Another method is pepsin digestion. Pepsin is an acidic protease that preferentially cleaves peptide bonds at the N-terminal side of aromatic amino acids, particularly phenylalanine and leucine, under acidic conditions. In the context of IgG, pepsin cleaves near the hinge region but closer to the Fc portion compared to papain digestion, resulting in the generation of Fab2 fragments. Next, we have physin digestion. Physin is a cysteine protease extracted from the latex of the fig tree and exhibits specificity towards peptide bonds adjacent to amino acids with bulky hydrophobic residues, similar to papain. In IgGs, physin can generate either Fab or Fab2 fragments, just depending on the concentration of cysteine activator used. Antibody fragments can be used as secondary antibodies in immunoassays for diagnostic applications. Fab fragments are particularly useful in double staining experiments as they can block background signals from primary antibody binding. Fab2 fragments are useful to avoid non-specific binding between Fc receptors on cells or to protein ARG. Due to having two binding sites, they can also bind and precipitate antigens. The three common immunoassays that these fragment antibodies can be used in are immunohistochemistry, immunofluorescence, and flow cytometry. Immunohistochemistry involves visualizing protein distribution in tissue sections using antibodies tagged with enzymes or fluorescent dyes. In immunofluorescence, one identifies protein localization through fluorescently labeled antibodies. In flow cytometry, antibodies conjugated to fluorescent molecules specifically bind to cell surface markers or intracellular proteins, allowing for the analysis and sorting of cells based on antigen profiles. Besides diagnostics, these antibody fragments can be used for therapeutic applications. Fab fragments are used in therapeutics such as antibody-based therapies for neutralizing toxins, targeting specific antigens, or modulating immune responses. They are engineered for enhanced tissue penetration and reduced immunogenicity, making them suitable for conditions like autoimmune diseases, cancer, and infectious diseases. One example is idarucizumab, which was approved by the FDA in 2015. It is a monospecific Fab targeting dabigatran and used to reverse dabigatran-induced anticoagulation for emergency surgery or in uncontrolled bleeding. Another example is ranibizumab, which was approved by the FDA in 2006 and is a monospecific Fab targeting VEGF-A and treats macular degeneration. Fab2 fragments may potentially be employed in therapies targeting cell surface receptors, tumor antigens, or infectious agents, providing improved potency and specificity. The only approved example we have is metuximab I-131, which was approved in China in 2007. It is a radioactive iodine-labeled anti-CD147 Fab2 monoclonal antibody to treat liver cancer. Fc fragments play crucial roles in therapeutic applications involving antibody effector functions, such as antibody-dependent cellular cytotoxicity, ADCC, complement-dependent cytotoxicity, CDC, and immune complex formation. They are utilized in antibody-based therapies for enhancing immune responses, eliminating infected or cancerous cells, or modulating inflammatory processes. One example is glofitamab, also known as Columvi, which was approved last year in Canada and is a bispecific targeting CD20 and CD3E. It treats diffuse large B-cell lymphoma. Another example is envafolimab, also known as Enwada, approved in China in 2021. It is a monospecific antibody targeting PD-L1 and treats microsatellite instability high or deficient mismatch repair advanced solid tumors. Biointron provides a wide range of services, including antibody discovery, expression, and optimization. We can take your gene sequence to purified antibodies in only 2 weeks with our antibody expression service. Our high-throughput platform enables the rapid expression of recombinant antibodies in HEK293 or CHO cells. We guarantee the delivery of 100 micrograms to 100 grams of recombinant antibodies. Remember, no antibody means no cost. Biointron is also a well-recognized leader in VHH antibody discovery and production. With our self-owned alpaca breeding farm, we offer a 1 alpaca per 1 project commitment and guarantee the delivery of over 20 unique binders, high diversity, and large capacity. We can also provide bispecific antibody production, afucosylated antibody production, single B-cell screening, hybridoma sequencing, antibody optimization, recombinant protein expression in the mammalian system, and CHO-K1 stable cell line services. Thank you for watching, and comment with any questions or topics you'd like us to cover next. Don't forget to like and subscribe for more.